good. Additional phage biodiscovery, that's done that. So we work on mainly with bacteriophages, which are viruses which target exclusively bacteria. And that is good because we can potentially we have a solution against AMR, multiple resistant bacteria. There's already what's happening now? Don't touch.
I'm trying to.
This is going to take a long time. Oh, okay, yes. So there's been.
It was just delayed, I think.
Recent papers on how actually phage therapy can go in now and save the world and animals and so. But also in a kind of hologenomics point of view, phages are the ones that actually keep the bacteria in kind of shape in a balance. And they are ancient, and they are the most abundant organisms on our planet. So there's estimated there's 10 up to 31 phages on Earth. And one can imagine what 10 up to 31 is. If you take all phages on Earth, line them up, you get a bridge which is 2 million light years far out of our galaxy. So they are the most abundant organisms on the planet, but also the least understood ones. And that is because they are so genetically diverse. If you look at bacteria and all the different phages attacking this bacteria, one would imagine how much similarity are between these phages. Turns out that Salmonella shares more genes with us human than many of these phages together. There's no similarity in the DNA level between them. There are no marker genes, let's say. Even if you start sequencing bacteria like Salmonella, you see all the genes in green are the ones we can predict the function, get an idea. All the red ones are the ones we can't predict. If you look at phages, it's completely opposite. Most phages we sequence, we have no idea. We see the proteins, but can't really understand what it is. This is because we have enormous genetic diversity. So, and those are, bioinformatics is mostly based on sequence alignment. You put them together, and that is how most of our tools work. So, what do you do when your sequence or genome doesn't match anything? This is the basic part of our group. So, this is the 6 different topics we're working in the group, and the biggest is machine learning. We're building a lot of different machine learning tools, not based on sequences, but based on other stuff to circumvent this lifestyle predictors, function predictors, and so on. And these are the people that have been publishing the group or building predictors. I'm going very, very fast over different things. If you want to know more, ask the people which are like this focus here. So the basic part of the machine learning is that we want to recognize the stuff which is not recognizable because there's no secret similarity. For that, we use machine learning. Then does this work? No. A little bit. Yeah. Then using kind of a variant of transcriptomics to be able to actually categorize the different phage genomes, categories, and then using knowledge graphs to compare different genomes. So, and also focus on AlphaFold. We're using a lot of predictive structures to actually dive more into the understanding of the proteins. So the machine learning, we are not doing based on sequencing. Most machine learning is done on images or sequences. We are moving into feature space, calculate a lot of different features, and use that instead. If you have more questions, you can ask Nastia. She's the only one who's still here on the machine learning part. The transcriptomics, and we'll not talk a lot about it, it's just we're using different phases in the transcriptomics to be able to pull out what a phage genome, what a phage is actually prioritizing, what kind of resources is it prioritizing to infect bacteria. The more interesting part is when we're just being able to start to compare different genomes which we don't know anything about. These were in the phage cloud. So these are all the circles are phage genomes, phage genomes. The size of the circle is proportional to the size of the genome. And you can see already that they're clustered by if they're similar. So you have a lot of different clusters, and they're the same phages or same family, but there's no similarity to anything else. And color is by kind of different taxonomy. What we want to do is to put all the different metadata where we know the experimental stuff about kill efficacy, about how it works together with antibiotics or not, because you can use antibiotics as it is. You can use antibiotics in cocktails, so they work together. You can use phages, similar, but you can also phages in cocktails to attack and prevent that the resistance comes up. But you can also use phages together with antibiotics. So all this information we want to put in on the knowledge graph, and being able then to transfer information we learn from one organism to another. So here, on the left, we have this phage knowledge graph on Salmonella. We have a lot of different experimental experiments about the data on Salmonella. You see all the Salmonella phages. You see all the genomes which are in yellow. These are the ones which we have used in phage therapy experiments and trials in the UK, and we know exactly how good they are or bad, and how good they are if you combine them. So the whole idea of using knowledge graphs is to put in all different kind of metadata, learn from one model organism for Salmonella how the phages working here, and then with machine learning transfer this information to any of the others, Streptococcus, Klebsiella, without actually having to do as many experiments as would be needed. So it works with that. So you build a knowledge graph. Here's a very simple one. You start getting this kind of metadata in. We create a knowledge graph which looks already complex. This is only 10 phages, but how they connected in the different data types. And then we want to do that on the million of phages we have now. This is also why Ben is kind of pushing. We need more servers, we need the memory, because this takes a lot of compute power. And another part we put into that information is those can move from sequence space into structure space. I guess most of you have heard about AlphaFold and ESMFold. We have been predicting 4 million phage proteins. This AlphaFold and ESMFold built our database, file database, and then started looking, can we actually use, is there any more information we can get out of that? And looks very good. So if any more questions, we're only sitting here. If we just look at one kind of protein from a phage and look at sequence, and then we just do normal blast. Here, that's the phylogenetic tree of all bacteria. And we blast, we only get hits against other phages attacking similar organisms like Salmonella, E. coli, all this the same usual suspects. If we jump into the structure space and do the same, suddenly we get actually hits against the whole phylogenetic tree. This is great because now suddenly we can actually increase our prediction. We can increase function prediction of what we previously couldn't do. So all these different tools we can apply in different very applied projects. So this is kind of interesting. We were a purely bioinformatics group, but all the recent rounds are very applied. So it's this kind of new for us. But so we were working with human health, trying to provide different algorithms to optimize. If you have a lot of data, a lot of different phages and antibiotics, how do you put them together in the best combination that they get the most effective phage cocktail? And also the recent one, I don't know how many of you have heard about necrotizing fasciitis, the flesh-eating bacteria, very nasty. If you get the infection, it eats 1 centimeter per hour of its own flesh. So it's here you can't, with a long time here, we work on getting phages and phage-derived therapy products. And if we raise Thomas, William, Peter, Thomas is here. Yeah, so more questions to that. We're also working in the animal health, trying to save the Norwegian salmon industry because they have a huge infection of Pasteurella. We're also the Chinese duck and chicken industry to actually predict and find phages to use instead of antibiotics. These are last year and things working that. And we are having a lot of trips in the rainforest for 2 reasons. People usually find phages in sewage and something else. We choose rainforest, much nicer to hike in that. But also there's another reason that's from the machine learning part. If you look at all the phage genomes which are available, you can see that 8 bacterial hosts are making up more than half of all the phages which are known. That means any one of you work machine learning knows that's a huge bias. So if you create a really nice predictor, most likely it will be a predictor that's really nice on Mycobacterium or E. coli, but not on the rest because you have such a strong bias. What we want to do is we're going around first, pick samples, try to find phages from regions where there are no humans have been. So we get a lot of salmonella phages, most likely because the monkeys are pooing everywhere. We're also using phages for tracing primate evolution. There are some megaphages which are 500, 600 kb, and they're very specific to the primate gut system. We're trying to figure that out. Then we have the recent biggest project we're running now with the whole group is we can actually find very variant phages hidden somewhere, which is where they haven't been found before. This is a collaborative effort of the whole group, and hopefully we'll, in a month or so, we can talk more about that and the whole idea and the paper. And then, of course, GLOBE is an ancient DNA institution. So we are looking, can phages be used as winners in the past? And the main idea is we see the phage, the virus, has a capsid. In the capsid is the DNA. The DNA is protected. We don't know for how long. We know at least 60 years, but is it protected 100 years, 1,000 years, million years? We don't know yet. This is what INADA tries to find out. And also, if it's protected for a long time, then we can actually get a glimpse on what diseases have been around, what kind of bacterial fragments we can find. All the published tools, as all the tools, machine learning tools we've been publishing, you can find on our predictor website and Phage Compass. And if you have any more questions to different people, these are the people in the group, which are the basics of all the different projects. Thank you.